So we'll be starting with the last part of my syllabus, that is the gene regulation part. And as usual, I have preferred iGenetics and Benjamin Pierce, and some pictures are of broker genetics analysis. So we'll move on to the Operon concept. Now, we have already done this particular picture when we studied the translation machinery. So this is how a monocistronic gene looks. If you remember, we have a common promoter element, what we studied, just remember that. We have a common promoter element. Then we had an open reading frame. This is what we have studied already. And then we have a terminator element. So at the promoter element, the RNA polymerase recognizes the promoter sequence, binds with the promoter sequence, and then starts transcribing the open reading frame, and then leaves the terminator part. When the open reading frame is transcribed, if you remember, there is a 5' untranslated leader region and a 3' untranslated trailer region. And the mRNA is then translated by the ribosome machinery, starting with the start codon AUG and ending with the either any one of the stop codon UAA, UGA, and UAG. This is what we know. Now, if I look into this particular gene, which I have showed you in the screen right now, you are seeing that there is another DNA sequence that has been inserted in between the promoter region and the coding region and is named as the controlling site. So this is a new concept. That is, in cases of some genes, it has been seen that there is a promoter element with the polymerase seeds, and there is a coding region. So this is the region from where our mRNA synthesis will start. But just preceding this coding sequence is a small DNA sequence, which is called as the controlling site, or at times named as the operator. So when we are talking about gene regulation, this particular controlling site is now providing an important controlling function over how the coding region mRNA will be transcribed. So when we are entering the topic of gene regulation, the first thing you have to know is that certain genes have a promoter element and a coding element. In between the promoter element and the coding region is a small yellow color sequence in the picture that is as is being shown is the controlling site, and then the terminator region. Now let us come to another concept, the concept of regulated gene and constitutive gene. In a cell, all genes are not constantly transcribed. Certain genes are regulated. These genes are activated only when the cell needs their product. Whereas if there is no need of that particular product, the gene is switched off. So these genes, which can be controlled, they can be switched on when the need arises and can be switched off when the need there's no more need of that particular protein anymore. These category of genes are known as regulated genes. With respect to regulated genes, there is another concept that is automatically that comes up is the concept of constitutive genes. Now, constitutive genes are genes, all organisms have a large number of gene products whose products are essential to the normal functioning of a growing and dividing cell, no matter what the conditions are. So certain genes have to be constantly activated. They need constant activation. They are not controlled by any switch. They are constantly activated in our cell. For example, the genes that codes for the enzyme needed for protein synthesis, or the genes that code for enzymes needed for glucose metabolism. That is the enzymes used in the glycolysis pathway. These proteins are constantly needed by the cell. So these genes are constantly active in the background. So these genes are known as constitutive genes or housekeeping genes. What picture you are now seeing is that of a regulated gene. This is a monocistronic RNA, mRNA. This is the monocistronic concept of a monocistronic gene, of a monocistronic picture of a regulated gene. So there is some sort of regulation being exerted as to when the mRNA will be produced and as to when to stop the mRNA. So when we are shifting from whatever we studied in central dogma, this is the additional part that we now realize that genes can be of two types. One is regulated, activate them only when the protein is required, deactivate them when the protein is not required. And the picture of the gene that has been currently shown on the screen is a picture of a regulated gene, where you are seeing that there is an additional yellow colored DNA sequences present, which to some extent will provide some sort of controlling or will exert some sort of controlling role onto the mRNA that will be transcribed. If we move to the next, so this is a monocistronic gene. Why monocistronic? Because there is a promoter element. On this promoter element, the RNA polymerase will bind, and you can see there will be only one mRNA produced from this coding region, and that single mRNA, so it's monocistronic mRNA, that single mRNA is producing only one protein. So this is a monocistronic mRNA. Now let's move on to the concept of another concept which we had touched when we had did translation, is a polycystronic mRNA. If we look into the structure of a polycystronic mRNA, so you see this is a polycystronic mRNA. We have a common 5' untranslated leader region, a common 3', I have again lost my cursor, just a second. A common 3' untranslated trailer region, and you can see that the first start codon, so it's starting from here. The ribosomes are identifying the shine Dalgano. The ribosomes are binding with the shine Dalgano. Formation of the 30S initiation complex followed by the 70S initiation complex, and start of translation from here. And this part of the mRNA is translated. And if you remember, I had said that there are some overlaps. So it's coupling of the translation. So in this overlap region, we have a sequence like this, which is the stop for the blue color part, and AUG is the start for the pink color part. So you can see the ribosome, after translating the blue color part, they release the polypeptide. So the polypeptide is released. This polypeptide is the beta galactosidase enzyme. It's an enzyme. We'll come to that name of the enzyme later on. It's an enzyme. And then the ribosome slides this part and again holds this pink color part. This pink color, it translates the pink color part. When it comes to the end of that pink color part, it releases the polypeptide. This polypeptide is again another enzyme. Again, it slides. So it has stop and start signals. And this is sort of a very light violet. It translates the violet color part and finally comes to the stop codon. The ribosome is released, the polypeptide is released, and we get a third category of enzyme. So you can see this single mRNA has the information of 3 different enzymes juxtaposed with each other. It has a common 5' untranslated leader region, and a common 3' untranslated trailer region. Since initially when the concept was given that one gene is equal to one mRNA and one gene is equal to one cistron, as you can see, the information of 3 enzymes are present here. So there are 3 cistrons present here. And hence, this is a polycystronic mRNA as compared to the first one I showed, where there is information of only one polypeptide was there. So that was a monocistronic mRNA. So this is where we developed the concept of polycystronic mRNA and monocistronic. So when we have developed the concept of what is a monocistronic mRNA and a polycystronic mRNA, just a second. Let's move on to the next part. Now let us come to the concept of Operon. So if you look into the concept of Operon, just now I said that in certain genes which are regulated, that is, they are activated only when their product is required. Otherwise, they are not activated. I showed you there was a stretch of DNA sequence present in between the promoter element and the open reading frame or the coding sequences. Okay. So as you can see in this particular picture, let's concentrate on this particular gene, which is a polycystronic 1. It has a common promoter element, as you can see. It has a terminator region at the end. It carries the informations of gene A, gene B, gene C. So we have an open reading frame starting from here, or the coding sequence starting from here. So we have a coding sequence starting from here. So we have a coding region starting from here. And just prior to the coding region and between the promoter and the first gene, you can see there is a small area which is, as I said, the controlling region, which I showed you in the first picture. So the area that acts as a controlling region. This area, in case of the Operon we are going to study, is known as the operator. So the operator, this DNA sequences present in the operator will somehow control how much amount of gene A, B, and C will be transcribed. How is this possible? So now let's look into the mechanism of how an operon works. When we talk about an operon system, it has been seen that most operons have a partner gene. So as you can see, this is the operon. I've again lost my cursor. Wait, just a second. This is the operon part. And there is a partner gene which is present a bit upstream from the original operon. So imagine a bacterial DNA, so chromosome. It has this lac operon. So this operon, we will be studying the lac operon present. And just further upstream of the lac operon, there is another gene which is known as the regulator gene, which is a monocistronic gene, which is also does not have any operator element present in it. So it behaves like the one we have studied constantly. So when we look at this regulator gene, you see this regulator gene has a promoter element, the coding region, and a terminator element. Simple, that's what we have studied. It's a monocistronic concept. It has a promoter element, the coding region, and a terminator element. Initially, what happens? This gene is transcribed to give us a mRNA, and then the mRNA is translated to give a protein product. This protein is known as the regulator protein, and in most books it has been referred to as the repressor protein when we study the lac example. So this is the regulator protein. So this is a once more monocistronic gene. The polymerase sits on the promoter, codes the, transcribes the regulator mRNA. The regulator mRNA is translated to the regulator protein. It is also known as the repressor protein. Now, the moment the repressor protein is translated, what happens? This repressor protein is a DNA binding protein. That is, this protein is capable of binding to DNA sequences. Not only that, it can also bind to certain molecules, which can be carbohydrates, which can be amino acids, and these molecules are known as the inducer molecules. So you can see it has two binding sites, region 1, region 2. This is schematically, this picture has been shown. So the regulator protein or the repressor protein that we are seeing is being transcribed from this monocystronic RNA has two binding sites. One can directly go and bind to the operator region, and the other, if the need arises, can bind to an inducer molecule. Now imagine what happens is that in normal condition, this gene is a constitutive gene. That means its product is usually continuously produced. And once this product is produced, what it does, it goes and binds with the sequences of the operator which it recognizes. So it goes and binds with the operator region. Because it recognizes the sequences of the operator, it's a non-covalent bonding, but it goes and binds with the operator region. The moment it will go and bind with the operator region, now see what the problem will arise is that the RNA polymerase is a big enzyme, if you remember. So it will come and sit here. That's the crab's claw. And it is supposed to transcribe these downstream elements. But in front of it, this regulator protein is sitting like a block. So it cannot move ahead. So it opens up. Since this regulator is present in the operator region, it cannot proceed further. And so what happens? It opens up. Now, so in normal situation, what we are seeing, that this regulator goes and binds with the operator and provides a block, just a second, provides a block. And since it blocks this portion, even if the polymerase comes and sits here, the polymerase is unable to move ahead of the block. Okay, so the polymerase opens up. But there is always a but. The but is that whenever we provide a small inducer molecule to the system, as I said, if I provide a small inducer molecule to the system, let's change this color. In color, let's change, make it blue. Okay. So the repressor is sitting there, fine. Now we have added an inducer molecule to the system. As I said, this protein has two binding regions. One with which it binds with the DNA, but the other part is free. So when this inducer molecule is present in the system, it goes and binds with this regulator element. The moment the inducer molecule goes and binds with the regulator element, there's a conformational change in that element. The conformation of that molecule changes. And the moment the conformation of the molecule changes, what happens? The regulator cannot remain bound to the operator anymore, and it is forced to open up. So the inducer goes and binds to the regulator, and the inducer regulator interaction brings about a conformational change in the regulator, and the regulator opens up from the operator region. Now imagine the situation. Now if polymerase comes and sits here, there is no more regulator in front of it. So it just moves past the operator, doesn't translate the operator. No part of the operator is translated. There's overlap between the first gene and the operator. The start codon is sitting here. Sorry, for the polymerase to synthesize mRNA, it starts from here. It starts from here. And you can see the mRNA is synthesized. And correspondingly, since there are three genes here, three different products are produced, and 3 different products are then used in another pathway to metabolize a particular substance. So this is basically the operon concept, where the operator element present in the DNA between the coding region and the promoter actually controls the transcription, downstream transcription of gene A, B, and C. If the operator is bound with its regulator, no transcription will occur. If the operator is free from the regulator, the transcription will occur. So this is a summary of the operon we will be studying. And there are 2 categories of operon in your syllabus. The one I explained briefly is that of the inducible operon, the lac operon. There's another operon in your syllabus, which is known as a repressible operon. We'll come to that later on, but first we'll get into the details of the lac operon. So this was a summary of our inducible operon. I gave you the summary of the lac operon, where we are getting an active regulator product constantly, which can bind with the operator. In its bound form, it prevents transcription. But if the regulator binds with the inducer and conformational change occurs, then it's free, the operator is free from the clutches of the regulator, and the polymerase can now transcribe gene A, B, and C. So now from the previous figure, as I showed you, that I almost gave you the example of the diagram via which I explained the operon concept was that of the inducible operon. And one such inducible operon that you will be studying in your syllabus is the lac operon. As you know that lactose is a major carbohydrate found in milk. It can be metabolized by E. coli bacteria that reside in the mammalian gut. One problem of lactose is that it does not diffuse easily across the E. coli cell membrane and needs to be actively transported. So there is a permease present on the E. coli cell membrane, which actually transports the lactose. And this lactose cannot be immediately utilized by the E. coli. For the E. coli to utilize the lactose needs to be broken down into glucose and galactose. So the lactose needs to be broken down into glucose and galactose, and this breakdown is carried out by an enzyme known as beta-galactosidase. So you can see the first thing is that the beta-galactosidase breaks down lactose into galactose and glucose. Another function of beta, so you can see beta-galactosidase is breaking down lactose into lactose and glucose. Another important function of beta-galactose is that it converts lactose into allolactose, which can also be broken down into galactose and glucose. So the one thing that we realized about this lactose is that it has difficulty in entering the E. coli cell, so it needs the help of a permease. Once it is present within the cell, it cannot be easily digested. It needs to be broken down into glucose and galactose. For that, it needs the enzyme beta-galactosidase. Further, beta-galactosidase also helps in conversion of lactose into allolactose. So this is the story of how lactose enters the cell and the pathway through which lactose is broken down and the enzyme required for the conversion of lactose to allolactose. Now, let's look at the structure of the lac operon, the lac gene. If we look at the lac operon, see, this is our lac gene. Now, if you draw similarity with the concept of what we have studied in our first slide, this is the promoter element. we have a small DNA element which will control the production of the downstream Z, Y, and A gene. So this is the operator element. And then we have 3 genes, Z, Y, and A, producing 3 different products. The first gene produces beta-galactosidase enzyme. So I just now explained what is the function of beta-galactosidase. It breaks down lactose into glucose and galactose. It converts lactose into allolactose. The second gene produces the enzyme permease. Just now we studied that permease helps in transferring lactose from the exterior to the interior of the cell. So that is the function of permease. It helps in diffusion of lactose across the cell membrane of E. coli. And the third enzyme gene is LAC-A, which produces the enzyme transacetylase. It's an enzyme that has some different function, but in case of Operon, what is its exact function is not yet known. So the 2 functional enzymes that works in Operon are beta-galactosidase and permease. One brings lactose from outside to inside. The other converts lactose to allolactose or break down lactose to glucose and galactose. And regarding transacetylase, it does not have any function with respect to Operon. There's one more thing I would like to say, emphasis on this side. See, when lactose is broken, it breaks down to glucose and galactose. If you remember, just when I gave you the concept of constitutive genes, I said there are certain genes which are constantly active. That included the genes which are involved in the glycolytic pathway. So when you have glucose in the cytoplasm, you do not need a regulated gene. The enzymes for glucose metabolism are constantly present in the cytoplasm. And hence, from the perspective of E. coli, if you have both lactose and glucose outside the cell, so E. coli will prefer glucose over lactose because the Operon is a controlled operon, but or is a regulated gene. But glucose gene is a constitutive gene. So the metabolizing enzymes for glucose are constantly present. So it will prefer that glucose will enter and it will digest glucose because it is less energy consuming also. See, when it takes in lactose, it cannot digest lactose. It breaks down lactose into glucose and galactose. So there is a requirement of ATP here. So if it brings in lactose, it has to break down it, providing energy to finally get to glucose. So if you give glucose and lactose to E. coli prefers glucose over lactose. Why does it prefer? This is the explanation. First, the genes for glucose metabolism are constitutive genes. The enzymes are constantly present. Second, if glucose enters the cytoplasm, it can be immediately used. But and no energy is required immediately. But if lactose enters the cytoplasm, it has to break down lactose into glucose and galactose. So energy expenditure is required. So if glucose and lactose are given together, glucose is a preferred metabolite as compared to lactose. So we came to understand that glucose is preferred over lactose. The genes for glucose are constitutive. The genes for lactose are regulated. And we have also understood the concept of mono and polycystronic gene. And since this is a regulated gene, we have understood the concept of a presence of a regulator element that will in fact control how fast the downstream genes Z, Y, and A are being transcribed. So, and another part, so this is one part where I explained the Operon, and while giving you a summary of the Operon concept, I said that all operons usually have a partner gene. So this is the LAC regulator gene, which is sitting upstream of the Operon. You can see it's a monocystronic gene. It has, again, I've lost my cursor. It has a promoter element. It has a promoter element, a coding region, and a terminate element, this coding region produces the regulator protein or the repressor protein. So this is the structure of LAC control system. It has a LAC gene, and preceding the LAC gene is the regulator gene. The LAC gene is a polycystronic gene. The LAC regulator gene is a monocystronic gene. So with this, I end my class for today.